Okay, so in the interest of time, I might get started and people may join as we go through the introductions. So just to firstly start by saying that it's my great pleasure to welcome you all here today to the Physiological Society webinar, Shaping Our Respiratory System. My name is Dr. Fiona MacDonald and I'm a lecturer and researcher here in the Department of Physiology at University College Cork in Ireland. And I'm joined by my colleague, Dr. David Burns, who's co-hosting today's event. So today's webinar aims to examine how our respiratory system develops in early life and how environmental changes can influence respiratory control and neurodevelopment during that perinatal period. So we have a really exciting lineup of speakers that you can see today that are all leaders in areas of the control of breathing, neurophysiology, immunology, molecular microbiology, and paediatrics, and are really driving research at the frontier. So the researchers have really a shared aim in addressing unmet clinical need in modern pediatric medicine. And today our speakers are Professor Nino Ramirez from Seattle Children's Hospital and University of Washington in the US, Professor Jodie Waters, who's at the University of Wisconsin-Madison in the US, and Professor Debbie Bogaert, who's at the University of Edinburgh. So we'd like to extend our gratitude to the Physiological Society for hosting this webinar, particularly Becky Jones and Kathleen Oates for facilitating this session. Each of our speakers will have 25 minutes, followed by 5 minutes of questions. During that time, you're welcome to post and like questions in the Q&A section during and following the talks. And then once all of the speakers have delivered their talks, we'll open up the floor for an exciting panel discussion. So I'll just now hand you over to Dr. Burns, who will introduce our first speaker of today's event. Thank you.
Welcome everyone, and thank you very much for joining us. It is my pleasure to introduce our first speaker, Dr. Nino Ramirez, who is Director of the Center for Integrative Brain Research at Seattle Children's Research Institute and a Professor of Neurological Surgery at the University of Washington. His research aims to understand sudden infant death syndrome, or SIDS, and erratic breathing in children with Rett syndrome, epilepsy, and other disorders. Dr. Ramirez has made a significant contribution to our understanding of gasping. He has identified a novel post-inspiratory complex, also known as PICO, and subsequently put forward the triple oscillator hypothesis, and his group is currently examining how the respiratory network assembles a breath. Dr. Ramirez is the current editor-in-chief of the Journal of Neurophysiology, and it's my great pleasure to welcome him here today, and I really look forward to your talk, Nino.
Thank you very much. Really, thank you so much for the invitation, Fiona, and also David. And thank you for the Physiological Society. It's a great honor for us to speak here. And I'm also thankful for all the people that are joining us. It could be early in the morning for some and later for others. Let's see. I have to navigate my screen a little bit. Do you see my talk? David, is it okay? No? Is it okay?
Yeah,
okay.
Looks good.
Okay, cool. Well, thanks again. And I'm talking today about the network control of breathing, but also cardiorespiratory coupling. And let's start first with what is breathing. So you have on one hand, inspiration, which is really driven by activation of the diaphragm, followed by the exhalation, where you use the recoil forces of the lungs to exhale air, but you slow down this exhalation by activation of laryngeal muscles, but also the diaphragm, a phase which is also referred to as post-inspiration. Now, under metabolic stress, for example, when you're jogging, then you also recruit intercostal muscles during a phase of active expiration. But really, during most times, you breathe between these two phases of inspiration and post-inspiration. Now, these post-inspiratory phase is also used for vocalization. So while I talk to you, I take a breath and then take this air to vocalize. But also, this phase is there for swallowing, because you don't want to swallow during an inhalation. Now, as a first approximation, there are two microcircuits that generate these two phases here. On one hand, the well-known Pre-Bötzinger complex established in 1991 that is defined by neurons that are called the DBX1 neurons. It's a transcription factor. It's glutamatergic neurons. And then more recently, the so-called post-inspiratory complex, also defined by glutamatergic neurons, but they have a co-transmitter called acetylcholine. So I have cholinergic glutamatergic neurons. And if you look at this neurons here in red, they are differentiated from the motor neurons that are only cholinergic. Now, you can isolate these two networks in a so-called horizontal slice, where you cut through the entire ventral respiratory column in mice, and you can get these two activities at the same time, the inspiratory activity, the post-inspiratory activity, in an alternating fashion. Now, you can see also that these areas are interacting. If you stimulate the DBX1 neurons optogenetically and record from the post-inspiratory neurons, you can see they're inhibited. But vice versa, if you stimulate the piconeurons, then you get activation of this population activity in phase with post-inspiration, inhibition of inspiratory activity, and the reset of the inspiratory rhythm. This is in vitro, but this is also in vivo. Similar situation, activation of post-inspiratory activity, reset of the respiratory rhythm. So there are reciprocal interactions. Now, what you can do is you can cut the slices also transversely, and now you generate two independent slices, one which is active, the post-inspiratory complex, and the other, the pre-Bötzinger complex. And what you can see is there's differential sensitivity, for example, to norepinephrine, but also more strikingly to actually opiates like Damgo inhibits at 25 nanomole the pico area, but not the pre-Bötzinger complex. And this is shown here in the dose response curve, 25 nanomoles, and shuts down pico, but really barely affects the pre-Bötzinger. Now, this is important because we can use this to show that actually this activity is necessary and also sufficient. So when you record in vivo now, here you have here the post-inspiratory activity, then you activate this pico area optogenetically, you get this post-inspiratory activity, and you can see here also you reset the next cycle. So the next cycle comes with a delay. Now, if you now inject Damgo into the pico area to inhibit these neurons, you lose this post-inspiratory activity. If you now activate light, you don't get much activation, and you also lose the reset, which has been quantified here. So before a delay, that is gone in the presence of the shutdown of this pico area. Now, when Alisa Huff joined the lab, she's a postdoc, what she found is she reproduced exactly this finding. She activates the pico area. She gets post-inspiratory activity here in the vagal nerve, but also in the hypoglossus. But she also recorded from the submental complex, and the submental complex is not activated in this stimulation. But you can see that when she stimulated here during inspiration, actually the submental complex is activated very dramatically, and also the laryngeal complex is activated dramatically, and you have actually a swallow. So basically the same area can activate either post-inspiration or it can activate a swallow. And there was a clear phase relationship. So if you activate during inspiration, like done here, then you trigger a swallow with most likely. And if you activate during expiration, you activate post-inspiration, but not a swallow. Now, these stimulations were done by activating cholinergic neurons. And when Dr. Luis Oliveira joined the lab, he actually introduced a viral approach where you can activate now specifically cholinergic glutamatergic neurons using a so-called Cree-Flippo approach. And now you activate only these glutamatergic cholinergic neurons and not the cholinergic motor neurons. And you get the same result. You can see during inspiration, you don't activate post-inspiration, but actually the swallow. And then when you go later in the cycle, you activate the post-inspiration, but not the swallow. Of course, we're just beginning now to unravel the circuitry. We're now looking also in the collica-fuse. We're interested how it interacts with the pre-Bötzinger complex. Now, when we come to the pre-Bötzinger complex, of course, we know much more already. So this is defined by the so-called DBX1 neurons. These DBX1 neurons are sufficient for generating an inspiratory rhythm. Optogenetic activation here in vitro initiates an inspiratory burst. If you inhibit these neurons with another opsin done by the Del Negro lab in vitro, you can see you can shut down the rhythm. But what's very important is that these DBX1 neurons have a phase-dependent effect on breathing. So when you stimulate these DBX1 neurons during inspiration, when they're actually active, you slow down the respiratory rhythm. When you activate them during expiration, when they're actually not very much active, you speed up the rhythm. And this was initially puzzling, but it's explained by the refractory period of these neurons. And this is something very important, because when you stimulate these DBX1 neurons right after an inspiratory burst, you cannot initiate the next cycle. It takes several seconds before you can initiate another inspiratory burst. And this, of course, is very bad for an animal, because these animals are breathing at 1 to 2 hertz. So a refractory period of 4 to 5 or to 7 seconds sometimes is not good for rhythm generation. And the reason for this is that they have an intrinsic, but also a synaptic refractory period. So if you isolate these neurons, you stimulate them, you get a long pause of several seconds before these neurons become excitable again. And the more you excite them, the longer this refractory period, and shown here. So a strong stimulation, long delay, short stimulation, a weak stimulation, and this much shorter. So basically, the more excited, the longer the refractory period, the less excited these neurons, the shorter the refractory period. And this excitability of these neurons is now actually controlled by inhibitory neurons. So 40% of the inspiratory neurons within the Pre-Bötzinger complex are actually inhibitory. And these neurons are concurrently active with the DBX1 neurons, and they bombard them with inhibition. So you can record from iPSCs during the inspiratory burst, and that reduces the excitability of these neurons. And by reducing the excitability, you reduce the refractory period. And in fact, if you stimulate now these inhibitory neurons during inspiration, when they're actually also active, you can see that now you can increase the frequency. So it's very counterintuitive, but the inhibitory neurons are actually increasing the respiratory frequency. And this is done in vitro, this is done in vivo here. An alert animal, you can increase the frequency up to 300%, which is way more than the DBX1 neurons can do, even if you stimulate them during expiration. But what's very important is that the timing is very important here. So in this reset curve, everything below of this phase shift is in a phase advance, and everything above is a phase delay. And in blue are the inhibitory neurons. And you can see when these neurons are active, they can cause a very dramatic phase advance, and that overcomes this delay caused by the DBX1 neurons. And when we look at the activation of these inhibitory neurons, you can see that they're specifically activated only during inspiration, during this time when they've caused a phase advance. By contrast, when you look at the DBX1 neurons, these neurons are also active before the inspiratory burst, during a time when they also cause a phase advance. So when you think about this, you can divide this respiratory rhythm, the inspiratory rhythm, inspiratory emphasis, into 3 phases. You have the burst, during which the inhibition and the excitatory neurons are active together, followed by a refractory period when all these neurons are shut down. And then you get the DBX1 neurons that get activated and cause a phase advance for the next cycle that starts then with a burst. Now, the hypothesis is that this 3-phase inspiratory rhythm here actually builds the temporal scaffold on which now in vivo, then the respiratory phases are established, because we know that during this refractory period is exactly when the piconeurons are activated. We know that the Pre-Bötzinger complex inhibits the piconeurons, and actually when you stimulate these the Pre-Bötzinger neurons, then you cause a rebound in the piconeurons, and the piconeurons then cause a reset of the next respiratory cycle. And now these interactions, they actually become critical in the in vivo situation. And in order to explore this, Nick Bush in the lab actually introduced the so-called neuropixel recordings, where he can record from between 500 and 1,000 neurons at the same time. And what he does is he inserts this probe in vivo along the entire ventral respiratory column. And that allows him to capture lots of neurons at the same time. And this is just an example. These are like inspiratory neurons here, and these are expiratory neurons here. And he can do this now for 20,000 neurons. And what he finds is that, in fact, the inspiratory neurons are focused here within the pre-Bötzinger complex, but they're not only there. They go along the entire ventral respiratory column. And then as you go more rostral into area of Bötzinger complex and pico, then you get more expiratory neurons. But really, there's a distribution of this inspiratory and expiratory neurons. When you look at the activity, and each line is one neuron here, you can see here in red, there's a predominance of inspiratory neurons, and then comes the expiratory neurons. But there's a gradient between early and late inspiratory neurons. And then there's, of course, a gradient also here between the post-inspiratory neurons and then here the E2 neurons. So you have basically a gradient, but clear division between inspiratory and expiratory neurons. What you can now do is you do a cross-correlation analysis between the activity of all these neurons and principal component analysis. And what you find is very beautifully that during each cycle, this respiratory network undergoes this alternation between expiration and inspiration. So here you have the inspiration, and this is the post-inspiratory phase, and then comes the next cycle. And I show you this again here in this slide here. Here's your three-phase rhythm, the three-phase scaffold, and the network goes here into expiration, and then comes the burst, and then comes the post-inspiratory onset, this phase. And what you see is really that the stable point is not inspiration. Inspiration is pretty variable. Expiration is variable, but what is not variable is this inspiratory off switch. So in other words, the inspiratory off switch is the attractor, and that's where the cycle starts. And so coming back to this concept of small microcircuits, this is clearly an oversimplification, because when you think about it, the network is really distributed along the entire column. And you have the inspiratory and expiratory neurons with a bias towards expiration more towards the rostral area and inspiration towards the caudal area. And the idea of microcircuits really comes from the necessity and sufficiency criteria, where you can cut out these areas and then you can say, okay, this is sufficient still, and this is sufficient for inspiration. But really the network is functioning really as a whole. And in fact, when you look anatomically, this is also reflected in the anatomy, because the DBX1 neurons are not restricted only to the pre-Bötzinger complex. They form a column. But when you record in vitro, then the inspiratory activity is focused on the pre-Bötzinger complex, but it's due to inhibition, because if you remove inhibition, then now you start to recruit also inspiratory neurons more rostral, and you get this distribution along the entire column. This is relevant during hypoxia, because in hypoxia, you get a reduction of inhibition. So this is shown here, you get the shutdown of inhibitory neurons, and now the network actually expands rostrally and generates gasping. And because you have this huge synchronization and the recruitment of a lot of neurons rostral to the pre-Bötzinger complex, now you have a long refractory period, and the breathing is slow, the gasping is slow. Now, inhibition, and as it goes into this gasping state, these dynamics of the networks are changing. So during inspiration, expiration, you have this rotational dynamics between the inspiratory and expiratory neurons. Now, as you go into gasping later on, you can see that the network changes totally its dynamic, and now it becomes ballistic. So it's not dependent anymore on the interaction between the inspiratory and the expiratory neurons. Now it really depends on the pacemaker neurons that now are driving the gasp in a ballistic manner. Now, a major source of inhibition comes from the afferences. So as you remove vagal afferences, you also recruit the rostral areas, and you slow down the respiratory rhythm because of an increase in the refractory period. And so when we conclude here, the network for breathing is really dynamically regulated. Vagal afferences reduce this network to the pre-Bötzinger complex and allow for faster breathing. And then during hypoxia, you disinhibit this network. It's increasing the refractory period, and you have gasping, presumably also activated by the chemoafferences. Now, breathing is not working alone. So it also functions in interaction with the heart. So during each inhalation, your heart rate goes up, and then during exhalation, the heart rate goes down. This is a sign of a healthy coupling, It's a respiratory sinus arrhythmia. And one of the consequences is also the diving response, because if you held the breath hold, then the heart rate goes down, and you can see this here. Now, this coupling between the heart and the breathing is disturbed in dysautonomia disorders. And one of the disorders is Rett syndrome. So each time this child inhales, you can see the heart rate goes up. You have a coupling here, which is still healthy, but then the child goes into a breath hold. It starts like a diving response with a decrease, but now the heart rate escapes. It becomes very high and then continues even though breathing starts again. So there's an uncoupling of the heart and the breathing in this episode of breath holding. And during this phase, of course, the child will suffer from hypoxia, and the child will not only have one breath hold, but the child has several breath holds in a row. And this is happening during the wake state, and we call this intermittent hypoxia, severe intermittent hypoxia. Now, there are several disorders of intermittent hypoxia, like obstructive sleep apnea. You have also this intermittent hypoxia, familial dysautonomia, also characterized by these intermittent desaturations. Epilepsy is also a dysautonomia disorder, because during each seizure, you expose the brain to hypoxia. And if you have several seizures, you have intermittent hypoxia. A consequence of this is that the heart rate is increased. So this is shown for Rett syndrome patients. You can see in black in Rett syndrome children, the heart rate is increased compared to controls. And when you look at the heart rate variability, it's dramatically reduced. And we have a recent paper where we worked on this. So all parts of heart rate variability are reduced. Dysautonomia is also associated with sudden death. So you have sudden death in Rett syndrome, in familial dysautonomia, in mitochondrial diseases, in epilepsy, etc. So what are the mechanisms? Now, when you look at the Pre-Bötzinger complex again, so this is a transverse slice, here in yellow are the cholinergic neurons for the nucleus ambiguus that also drive the cardiovagal neurons. And right below this is the Pre-Bötzinger complex. And so basically during each inspiration generated by the Pre-Bötzinger complex, these cardiovagal neurons are inhibited, which means the heart is disinhibited and goes up. And this reciprocal activation is pretty striking and contributes to the sinus arrhythmia. Now, of course, you have also sympathetic drive. And this is work done by Malusa Amarante from Brazil. And you can see that in vivo, you can see the activation during inspiration of sympathetic nerve activity. But you can also see here some post-inspiratory activity, not as striking as the inspiratory activity. And this is driven in part by the pico area. So if you stimulate pico area, then you can also activate the sympathetic nerve activity. So what happens now, basically during each inhalation, you have a concurrent disinhibition of the heart by the cardiovagal neurons, and you have an activation of the heart by the predominance of inspiratory activity from the sympathetic nerve. Now, when you go into hypoxia and chronic intermittent hypoxia, you get an upregulation of HIF-1 alpha, and you get also signs of oxidative stress. So when you record from these cardiovagal neurons and you apply hypoxia, you can see the production of hydrogen peroxide. And as a long run, you have lipid peroxidation, sign of oxidative stress. Now, what happens to the neurons during this? So hydrogen peroxide inhibits this activity in these cardiovagal neurons. So you're losing this activation. So the heart rate will go up and the heart rate variability goes down. Now, this effect is, of course, not the only thing that happens, but it contributes to this. And there's beautiful work done also by Benedito Machado and his groups, and David Moraes, and they kind of show how basically the activation of the sympathetic and parasympathetic drive is changed after chronic intermittent hypoxia. So when we looked at the sympathetic drive after chronic intermittent hypoxia, what we see is that now you get also a dramatic activation of post-inspiration and also E2, really what also Dr. Machado showed with his group. And as a consequence of this, you're losing this predominance of inspiration because now you're activating the network also during the other phases. So when we look at now at this heart rate variability, you lose the disinhibition from the cardiovagal neurons, and you're actually also activating sympathetic activity during inspiration and also exhalation, which certainly contributes to the dysautonomia. Now, in addition, the Pre-Bötzinger complex is also affected. So chronic intermittent hypoxia reduces the excitability. And as a consequence of this, these neurons don't synchronize anymore so well. So you get these failures of synchronization, where you can see now the population activity shows this very dramatic amplitude variation. You see this in vitro, but you can see this also in vivo. You can see the variability from cycle to cycle in the amplitude, because now you lose synchronization between these inspiratory neurons. And this is a big problem, because the hypoglossus is activated during each inspiration by the Pre-Bötzinger complex. This is shown here, that during each Pre-Bötzinger complex, the hypoglossus is activated. And what happens now if you have chronic intermittent hypoxia, you get these hypoglossus apnea, a mismatch between the inspiratory drive to the diaphragm and the hypoglossus, and that causes a pharyngeal collapse and contributes to obstructive sleep apnea. So what I showed you basically is that after chronic intermittent hypoxia, this excitability decrease leads to a desynchronization and a huge variability in the amplitude, a variability in the drive. You lose also cardiovagal coupling. And this is all driven by reactive oxygen species. So if you apply MNT and PYP, it's a scavenger, you lose this variability, and you reinstate actually the transmission to the hypoglossus. So in summary, what I showed you is that the cardiorespiratory control is basically all happening here with this interaction between the breathing and the cardiovagal neurons in the hypoglossus. And this is all coordinated and sensitive to hypoxia. I showed you that in hypoxia, the network can dynamically be regulated, and all these changes seem to be driven by reactive oxygen species. Now, it is tempting to hypothesize that ROS activation by intermittent hypoxia is only one of the conditions that can be involved. And we know that reactive oxygen species is also activated by heavy metals like lead, air pollutants, and also infections. So infections also drive reactive oxygen species. And I have here some very anecdotal evidence from the internet, where a patient posted the activity of the heart rate variability after being infected with COVID, and you can see this reduction in the heart rate variability. But more controlled, this is work done by Mark Wainwright in our hospital. He's studying sepsis patient, and you can see here this heart rate variability quite dramatically. And in this patient at 6 p.m., this patient shows a sudden drop in the heart rate variability. And then at 6 a.m. in the morning, 12 hours later, the child suffers from cardiac arrest and then expires. So basically, by looking at the heart rate variability, you can predict this event. And this will be, of course, clinically extremely important going in the future. So let me summarize what I told you today. So every breath is really generated by a dynamically controlled and distributed network that is distributed along the entire ventral respiratory column, with the predominance of inspiratory neurons in the Pre-Bötzinger area, but really it goes up in the expiratory more rostral, where you have the Bötzinger and also the Pico area. Then the inspiratory off switch is really the attractor, the only stable point in the breathing rhythm. And so you can say that really the breathing cycle begins with post-inspiration, and then basically goes through expiration and then the initiation of inspiration. Now, every breath is also coordinated with sympathetic and parasympathetic control of the cardiovascular system, and the rhythmic orchestration is now differentially altered in disease, for example, in chronic intermittent hypoxia. Now, many of these changes are driven by reactive oxygen species that seem to disturb these networks and dissociate these networks, and then lead to a disease. So in the end, I want to thank my lab. We have here Nick Bogaert, who does a lot of work on opiate respiratory depression. Then this is the team of Malusa and Alisa working on swallowing chronic intermittent hypoxia. We have Nick Bush working on the neuropixels, and then Luis from the Takakura lab, he works on the network analysis of this network. And then I have other very cool people, but I had no chance to talk about this. But I want to thank also the NIH, and thank you very much for listening to my talk. So I stop sharing.
Thank you, Nino, for a very elegant talk with a great range of techniques. We're open for questions.
David, I think you're muted, or is it? I don't hear you.
I can hear you okay.
I hear you.
Okay.
Okay. We have a question from Duncan Banks. Very interesting talk. What neuronal types are most sensitive to mu opioid agonists such as fentanyl?
You know, we have a developing story where actually the tonic neurons play a big role in this. And this is just a little surprise, but the pico neurons, the post-inspiratory neurons are clearly more sensitive than the inspiratory neurons. So there's a differential sensitivity. I also want to mention that, you know, I did not talk about colic of fuse, but obviously colic of fuse plays also a big role, and as shown by other groups.
Thank you, Nino. We have another question from Jim Dukers. How does hypercapnia influence the system? So I guess you mentioned hypoxia, but do you have any insight as to how hypercapnia or even dual stimulus of hypoxia and hypercapnia can influence the network?
First of all, hi, Jim, and sorry, you know, like we plan to do this, you know, like the pico area sits right at the RTN and we haven't done it yet, but I could imagine that there are big interaction between these areas, but we have not studied it.
Brilliant, thank you, Nino. I think in the interest of time, we might move on to the next speaker as we're running a little bit over time, but we'll be able to come back to Nino again as part of the panel discussion. So looking forward to fleshing out some more of Nino's talk, I guess. So I'm going to hand you over to Fiona, who's going to introduce our next speaker.
So thank you very much, Dr. Ramirez. So next speaker is Dr. Jodie Waters. She's a professor at the Department of Comparative Biosciences at the University of Wisconsin-Madison. Dr. Waters' research group is interested in how fetal exposure to intermittent hypoxia during pregnancy impacts offspring brain development and function. So a key aspect of her research centers on understanding the differences between male and female microglial responses and how these sexual dimorphisms influence central nervous system function and neuroplasticity. So Dr. Waters, you're most welcome, and we're really looking forward to your talk. Thank you.
Oh, Jodie, you're muted.
You're muted. Sorry.
Here I am. Okay, can you hear me now?
Yeah, good.
Okay. All right, and now can you see my slide correctly?
Perfect.
Okay, all right. Thank you, David and Fiona, for the very kind invitation to come present today, and also to the Physiologic Society for putting on this webinar. I today would love to present to you some of the work that we've been doing looking at how intermittent hypoxia in mothers during their pregnancy impacts the respiratory system of their offspring. And before I forget to do the most important thing, I need to mention that all of the work that I'm going to be presenting to you today was done in collaboration with my colleagues, Professors Tracy Baker and Steve Johnson here at the University of Wisconsin-Madison. So the idea that chronic disorders in adulthood can be explained only by bad genes and an unhealthy adult lifestyle was challenged in 1990 by the British epidemiologist David Barker, who proposed the fetal origins of adult disease hypothesis that says that the intrauterine milieu is just as important as the genetic endowment in shaping the future health of a conceptus. And so he proposed a gene by environment interaction, an idea that suggests that adverse events to which the mother is exposed could reprogram the physiology of her fetus in order to make it better able to adapt to the environment into which it's going to be born. And so David Barker and his colleagues formalized this association between the intrauterine environment and later life disease risk by performing epidemiologic studies in adults that were born during the Dutch famine of 1944. And his group found a striking correlation between the low birth rate that was presumably due to the maternal famine and a risk for obesity and associated health problems like heart disease and diabetes in adulthood. And so this association between prenatal undernutrition and later life metabolic disorders has since been overwhelmingly confirmed in a number of animal models and other human studies. And we are proposing that the adult respiratory control system is one such system that can be influenced by that perinatal or intrauterine environment. So we now know, in addition to maternal malnutrition, that there are several maternal factors that can lead to the reprogramming of offspring physiology. For example, maternal stress or consumption of drugs or alcohol, poor diet, obesity, underlying health conditions like hypertension or diabetes and infection. And all of these maternal factors have been associated with a variety of diverse conditions, both in the juvenile or in the adult offspring. But one thing that all of these diverse maternal factors have in common is activation of the maternal immune system or maternal immune activation. And so we propose that another commonly overlooked inflammatory stimulus in the mother is sleep apnea during pregnancy. Sleep apnea, as you know, is increasingly common in the general population. And importantly, it is also common during pregnancy, such that about 15% of all pregnant women develop some form of sleep apnea by the end of their third trimester. And it's known that maternal sleep apnea in pregnancy has adverse consequences perinatally, so on the health of the newborn and the mother. But whether these adverse consequences extend into adulthood is incompletely understood at the moment. Interestingly, also, sleep apnea is heritable, and familial aggregation of sleep apnea is known to be independent of obesity. The risk for sleep apnea is about 2 to 4 times greater in relatives of individuals that have sleep apnea. And so while genetic abnormalities in the upper airway anatomy, of course, contribute in some cases, familial aggregation of sleep disordered breathing is apparent even without these overt craniofacial abnormalities, and therefore may be related to ventilatory control deficits, such as impaired neural control of upper airway during sleep. But neither the precise mode of heritability nor specific gene mutations that are involved in this to increase propensity for sleep apnea are known. Intermittent hypoxia and sleep fragmentation, of course, are hallmarks of sleep apnea and can occur many tens to hundreds of times per night. And so accordingly, intermittent hypoxia is a very profound inflammatory stimulus, and in fact can upregulate many of the same inflammatory cytokines that are known to contribute to offspring reprogramming in other models of maternal immune activation that we just talked about. So the underlying hypothesis for our work is that, similar to other in utero insults, we suggest that maternal inflammation induced by intermittent hypoxia during pregnancy can also cause long-lasting consequences to offspring respiratory function in a manner that involves chronic neuroinflammation. And so today I want to share with you a little bit of the work that we've done looking at neuroinflammation and neural control of breathing, and some of the initial molecular mechanistic studies that we've begun to undertake to better understand some of the adverse consequences of maternal intermittent hypoxia on her offspring. And so the model that we use is a rat exposed to chronic intermittent hypoxia in a chamber during gestational days 10 to 21. And so I want to note here that this model does not model the concomitant sleep fragmentation that is a hallmark in humans, nor does it model intermittent hypercapnia. So we exposed our rat dams to 15 episodes of hypoxia. We go down to about 10.5% for 8 hours during their sleep cycle. And we call this paradigm gestational intermittent hypoxia. And the control dams are exposed to normoxia, intermittent normoxia, during those same gestational days of exposure. And we call the controls then GNX, or gestational normoxia. And I want to mention here that the mothers, the pregnant dams, desaturate to about 85%, which is similar to what humans with sleep apnea experience. I also want to point out that the kinetics of the intermittent hypoxia exposures that we use here are designed to model the kinetics that are reported by Alan Pack's group. And so the day before birth, the dams are removed from the exposure system, and the pups are allowed to be born into normal cage conditions so that they're never directly exposed to IH. They only experience intermittent hypoxia in utero. The offspring are allowed to grow to adulthood. For the studies I'm going to show you today, somewhere in the zone of 3 to 5 months or so of age. And after that time, we study their breathing and their microglial inflammatory activities. And I'll come back to the microglial part later. I also want to mention here that in these moms that we have exposed to intermittent hypoxia, we have not noted any differences in the length of their gestation, in the number of pups per litter, in the retrieval time of the moms to retrieve their pups once we take the pups away, or in food consumption of the mothers. So one of the first respiratory function measures that we looked at in the offspring was whether or not they had apneas. We did this by plethysmography, and we categorized the apneas into 3 major types based on what we observed. So the first was a spontaneous apnea. And here I'm showing you an example of what we consider to be a spontaneous apnea, which we define as 2 or more missed breaths without an apparent cause. We categorized also the apneas as post-SI apneas if they occurred after an augmented breath. And we also noted that at times there was a series of apneas that occurred after an augmented breath that was separated by a normal breath or 2 in between. And so we called these post-SI trailing apneas. I also want to note that the data that I'm showing here are just in the males, because we did not observe any of these phenotypes in the female offspring. And so what we found was that the GIH male offspring had considerably more spontaneous apneas, but there was no change in the frequency of their post-SI apneas. But interestingly, with these post-SI trailing apneas, we did see a trend towards elevated levels of these post-SI trailing apneas in the male offspring. But one of the things that I want to point out here is that we don't expect every litter and every pup in a litter to be impacted by maternal intermittent hypoxia, because that's just not realistic. It doesn't happen in humans. But we believe that there's important information that we can derive from the variance in these data. So we parsed the data into what we called responders versus non-responders. And when we did that, And so we identified a responder as male offspring that had a spontaneous apnea outside of the 95% confidence interval in the normoxic offspring. And we found when we did this that about 67% of our male offspring fell into this responder category, which suggests that the phenotype is apparent in about 2/3 of our animals. And when we do this, when we do this, so within the responders, we also looked at the post-sci apneas, which still didn't change, but the post-sci trailing apneas did then become statistically increased in the male offspring. So if you just look at the responding offspring, the males have increase in spontaneous apneas and also in these post-sci trailing apneas. Okay, so then we shifted gears a little bit to look at another respiratory system output, and that is the ability for these offspring to express neuroplasticity in the context of breathing. And so plasticity is, of course, a fundamental property of a healthy respiratory control system, which allows the individual to adapt to a myriad of changes that the organism would experience throughout life, such as development, aging, gain or loss of weight, learning to coordinate breathing with other activities like singing, illness or injury, ascent to altitude, pregnancy, exercise, so on and so forth. And so the respiratory control system needs to be able to adapt to all of these different circumstances, and plasticity of the respiratory system then is one way that we can adapt to these circumstances. Plasticity typically occurs after an abnormal or repeated experience, and in our case, an experience such as recurrent apneas in individuals that have sleep apnea. And so work from Tracy Baker's lab has shown that respiratory neurons in normal rats are exquisitely sensitive to reductions in respiratory neural activity, even in the absence of changing blood gases. And so in rats that are anesthetized, paralyzed, vagotomized, and mechanically ventilated, the phrenic nerve is dissected and electrophysiologic output is reported. And what you can see here is a representative phrenic neurogram depicting a normal response to recurrent reductions in respiratory motor output. This image is compressed to see the amplitude of phrenic inspiratory motor output over time before and after 5 recurrent pauses in respiratory activity. Because these rats are mechanically ventilated, though, throughout the protocol, they do not experience hypoxia or hypercapnia when the 5 neural apneas are being delivered. And so as you can see, following each pause in firing, which I should mention doesn't need to be a complete reduction or a complete cessation in firing, or reduction is sufficient, the phrenic amplitude progressively increases. And this increase in phrenic inspiratory output is termed IMF, or inspiratory motor facilitation in response to inactivity. And so Tracy and her colleagues have also shown that the magnitude of this form of facilitation is directly proportional to the magnitude of activity deprivation, and that this results in a proportional reduction in CO2 apneic threshold that makes it harder to trigger the next apnea. So this form of plasticity, we hypothesize, is one that's necessary to prevent subsequent apneas. And so the studies that I'll show you here, we're using this form of compensatory plasticity as a readout for how the adult offspring respiratory control system is impacted by those gestational exposures to intermittent hypoxia. So the question that we asked then was whether or not this gestational intermittent hypoxia exposure impairs these normal compensatory responses to recurrent central apneas. And so what I'm showing you here now are representative traces of the phrenic neurogram, just like I showed before, in the normoxic control male offspring in black and the GIH male offspring in red. And like I showed before, this increase in phrenic nerve amplitude is what we are looking at in terms of plasticity that the control males express, but the GIH male offspring lack. Those data are quantified over here where phrenic inspiratory motor output in the normoxic male, that elevation is absent in the female or in the GIH males. And again, like I mentioned before, with the plethysmography data, we do not see any deficits in females. The control and the GIH females behave similarly. And so we know from our previous work, from our group and others, that neuroinflammation in adult animals undermines respiratory plasticity. And so we hypothesized that GIH offspring might have persistent or chronic neuroinflammation that could be undermining the ability for IMF mechanisms to be expressed. And so we tested or began to test this hypothesis by just looking at the expression of inflammatory molecules in male and female offspring, both in utero, so at gestational day 19, and then also in adult animals. And I'm just showing you one cytokine here, but this is true for several cytokines. You can see in red that the GIH offspring, both male and female in this case, do have elevations in interleukin-1 beta mRNA in this case. But if you look in the adult animals, the females no longer have that persistent interleukin-1 beta, but the males do. And so we wanted to test whether or not inflammation or chronic inflammation was contributing to the absence of plasticity in our GIH male offspring. And so to do this, we delivered spinally an anti-inflammatory drug called TPCA. This is a pharmacologic inhibitor of the NF-kappa B and STAT transcription factor pathways. And what you can see, like we saw before, the GIH male offspring do not have plasticity, but when we deliver this anti-inflammatory molecule over the phrenic motor pool, we can restore IMF in our GIH offspring, suggesting that local inflammation in the spinal cord is inhibiting IMF in our male offspring. And so because microglia are one of the more important cell types in the CNS that contribute to inflammation, and they have such important roles in neural function, we hypothesized that microglia might be the producers of this chronic inflammation that we observed in the adult brain. And to test this hypothesis, we delivered a pharmacologic drug called PLX3397, which is able to induce death of microglial cells. And it does so by inhibiting this receptor, the CSF1 receptor, whose signaling through this receptor is required for microglial survival. And so you can deliver this drug for 7 days orally. And as you can see here in this middle panel, the microglia, the majority of them will undergo apoptosis. This is immunohistochemistry looking at two different markers of microglial cells, IB1 and CD11B, and then their overlay. And you can see that after 7 days, the majority of microglia in this is the cervical spinal cord, are depleted. And one important and useful aspect of this PLX drug is that once you stop giving it to the animals, the microglia will repopulate the CNS. And so that's just shown here. This is 3 days after withdrawing the PLX drug from the animals. And so it allows us in the same animal to look to see what the consequences of microglial ablation are and then microglial repopulation. And so what we found was if we ablate microglia, this is the control, the GNX males, the absence of IMF that we normally see in our GIH males is now corrected when we take away microglia in the males. And over here, I'm showing you the females again. The females never do anything interesting over here. What was surprising to us, however, was that if we allowed the microglia to repopulate, the plasticity that was restored when we took away or abolished or ablated the microglia, the plasticity went away again. And so this suggests to us that whatever alteration is happening to the microglia as a result of that in utero exposure is held somehow in either the microglial progenitor population in the CNS or in those PLX resistant microglia. Not every microglial cell was depleted. And so that is interesting to us because it seems that repopulating the CNS with microglia in the context of this gestational intermittent hypoxia exposure is not able to correct the phenotype. Okay, so one other thing that we know about microglia from these adult animals is that their responses to a typical inflammatory challenge are also exaggerated. And so what I'm showing you here, I'm showing you a different inflammatory gene just so that you can see that multiple inflammatory genes are upregulated in these adult offspring. You can see in isolated microglial cells that if we challenge animals with LPS, gram-negative bacterial mimetic, you can induce an inflammatory response in normal, the control animals. What was interesting to us, however, besides the fact that there's an elevation in basal gene expression in the GIH animals, is that when you challenge the GIH animals with LPS, their response to that inflammatory challenge is much more robust. And so this suggests that the microglia in the GIH animals are actually primed. And one of the major mechanisms that underlies gene priming in immune cells, and this is data that we know much more about from peripheral immune cells, involves histone modifications. So in peripheral immune cells, gene priming or memories of a prior exposure to a pathogen are stored in the chromatin by virtue of epigenetic marks that are placed often in gene promoters or gene enhancers of those inflammatory genes as a consequence of that first exposure to pathogen. Those marks function to enable a more robust and a quicker response to that same pathogen upon re-exposure. And so you can see here that normally the chromatin is held in a closed conformation, especially around inflammatory genes normally. Upon first exposure to pathogen in this case, marks are placed on histone tails that open the chromatin and allow transcription factors to bind and then transcribe those genes. When you wash away the pathogen, the chromatin closes again, but those marks, those histone marks stay. And the idea is that because those marks are still present, when a subsequent challenge comes along with that same pathogen or even a different one, the chromatin is primed, if you will, to be more readily and more quickly responsive to that pathogen. And so there's a bunch of different histone modifications that can open and close the chromatin. And I'm just going to focus here today on the histone 3, lysine 4, tri and monomethyl marks. Those are activating marks, and they are typically found at gene promoters or gene enhancers. And so because microglia make up just a very small percentage of total cells in the spinal cord, it's actually about 5%. And we typically need millions of cells in order to sequence the epigenome. We turn to an in vitro hypoxia system where we could culture microglia, we could expose them to hypoxia and challenge them the next day with LPS and observe the same kind of priming responses that we observed in our GIH offspring. And so we did experiments like this with microglia in vitro, and we performed chromatin immunoprecipitation sequencing of the entire genome. And then also we looked by ChIP-Q RT-PCR at specific gene promoters that we see to be primed in our offspring. And so here is just an example output of some of the ChIP-seq data for H3K4 trimethyl marks in this case at one gene in the microglial genome in control animals and in hypoxic or animals, microglial cultures or hypoxic exposed cultures. And what we're looking for are these increases in the height of these peaks, if you will, which indicate enrichment of the H3K4 trimethyl mark in this case. And so we analyzed the entire genome this way and found that with hypoxia exposure, there were over 17,000 new H3K4 trimethyl peaks that appeared that were unique to hypoxia, that were not present in normoxia or that were not shared with normoxia. We performed gene ontology analyses to figure out, well, what are these 17,000 genes? And in general, they were either cytokine genes or they were metabolic genes. And so given that we are interested in microglial inflammatory activities, we started looking at the cytokine inflammatory genes to see whether or not the particular primed genes that we observe in our GIH offspring are present in these mice and whether or not they have enrichments in H3K4 or trimethyl or monomethyl marks. And so here I'm just showing you the results of a ChIP qPCR experiment where we looked for H3K4 trimethyl and monomethyl mark enrichment at various gene promoters. And what you can see is that at the INOS gene, at the COX2 gene, at the TNF alpha gene, there was enrichment for H3K4 trimethyl mark. We didn't see that at the IL-1 beta promoter, which, as I showed you in vivo, was one of those genes that was chronically elevated in GIH offspring, and that was primed. But we did find H3K4 monomethyl mark at the IL-1 beta promoter. And so we're working right now on a way to do ChIP-seq in vivo on microglia that are isolated from GIH offspring. We can only get about 50,000 cells when we do the in vivo experiments, and we need about 10 million to do these experiments. So it's a little bit of a technical challenge, but we're making progress. And the goal with those experiments is to help us identify whether or not similar epigenetic modifications that we can see in vitro are actually also occurring in our GIH offspring. Okay, so our general working model is that the intermittent hypoxia exposure of the moms during her pregnancy can somehow epigenetically reprogram those fetal microglia that cause them to be chronically inflamed, if you will, in a way that persists into adulthood, and that their inflammatory activities interfere with respiratory neuroplasticity. In this case, we're looking specifically at this homeostatic form of plasticity, IMF, in adult offspring, that because it's absent in these GIH offspring, we think gives rise or facilitates the increased apneas and hypopneas that we observe. And then lastly, and again, most importantly, acknowledgements. We need to thank everybody who has been working on this project, both in Tracy and my lab, but also in Steve's lab and in labs of our collaborators whose work I didn't have time to share today. Also, would like to acknowledge our funding and you all for your attention, even though I can't see you, and ask if there are any questions.
Thanks very much, Jody, for a really interesting talk. I have lots of questions. I might just ask one in the interest of time, and then we can discuss some of the broader questions later on. I was just wondering, have you measured oxygen in utero in the animals? And do you see a big difference or?
Yeah, that's a great question, and it's a really hard question to answer. And so, especially to measure, fetally with a electrode probe. I mean, that's really hard. And so, no, we haven't done that. But what we have done is use a hypoxia probe, which is a dye that we can deliver to the mothers that will, in the presence of hypoxia, change the conformation of that dye, and then we can detect that by Western blot. And so we're in the process of doing those experiments to determine whether or not the fetal brains in particular are becoming hypoxic.
Yeah, because I guess the question, there's an interesting question there, whether it's like information coming from the mother or whether it's, information generated from the actual fetus itself. Exactly.
Yeah, and inflammation, like maternal inflammation, can elicit these very same kinds of epigenetic alterations as well. And so actually I didn't have time to show those data, but we've also induced gene priming in our microbial cultures in vitro by delivering cytokines.
And have you measured cytokines in the maternal system?
We're doing that. We are doing that right now as we speak.
Thanks very much, Jody. I'll hand you over now to David to introduce the next speaker. Thank you.
Thank you.
Thanks, Fiona. Okay, so our next speaker is Dr. Debbie Bogaert, who's a professor of paediatric infectious diseases at the University of Edinburgh. Her research group focuses on investigating the physiology and pathophysiology of respiratory infections and inflammation from an ecological perspective. The ultimate goal of Professor Bogaert's research is to design new or improved treatment and preventative measures for respiratory infections in susceptible populations. And to this purpose, our research team uses a fully translational approach, combining epidemiological, molecular microbiological, immunological, and systems biology approaches to answer their research questions. So thank you very much, Professor Bogaert, for joining us. And I look forward to hearing more about your research during the course of your talk.
Thank you, David. Can you see my screen?
Yep. You're not in presenter mode yet. Yeah, perfect.
Yeah. Well, thank you very much. And thank you for the invitation to be part of this exciting webinar. Let me know if my upload is a bit dodgy, because then I will close my camera, okay? So I think we've heard today a bit about interactions between multiple organ systems and the respiratory system. And I think I'm gonna add an extra organ system, which actually is only, I would say, research for the last 15 to 20 years, which is the microbial community or the human microbiome. So I first want to give a brief introduction to just get everybody a bit up to date where we started and how we got where we are at the moment. So I think most of you by now have heard about the human microbiome. It's not a surprise anymore that our bodies are all covered on all outside and inside surfaces with a very diverse community of microbes. And it consists of about 37 trillion cells, which is even slightly estimated to outnumber our human cells. But if you think about the genetic capacity that microbiome has, it outnumbers our own genetic capacity by a factor 100. So it makes then all of a sudden sense that the functions that this microbial community elicit for our body are extremely important. The thing is that a single human microbiome does not exist. We speak about the human microbiome, but the microbiome will adapt on your body and in the surfaces and niches that it's inhabiting based on what it needs themselves, what environment it needs, and also what functions it needs to elicit reciprocally for our human body. So that also counts for the respiratory system. So if you think about the respiratory tract and the commensal microbes that are inhabiting our respiratory tract, what are these microbes supposed to do? Well, there's a couple of levels of effect that they have. So commensal microbes can directly inhibit or compete with pathogens for bacteria, for nutrients, but also by producing bactericidal and metabolic byproducts to really keep pathogens at bay. The other influence it has is by directly interacting with the mucosal layer that they are inhabiting. And by that interaction, it will stimulate mucosal barrier function, development, but also production of things like mucus and antimicrobial peptides. So, of course, these in return have an effect on pathogenic colonization and an equilibrium on the surface. And then there's a third layer that we know that the commensal microbes, albeit they are on the surface of any mucosal or epithelial layer, that they can directly interact with our immune system beneath, especially through dendritic cells, example, and thereby they can interact with our immune cell population. And if you talk about early life, which is the field I'm mostly researching, helping to get immune maturation going, and especially helping that balance, which is essential for healthy living, between tolerance when you're healthy and you're not supposed to produce an overwhelming inflammatory response to your environment versus having an appropriate inflammatory response when it's needed in times of an infection, for example. Okay, so what we do know, we know that already for many decades, is that the upper respiratory tract, especially the nasopharynx, the back wall of the nasopharynx, is basically the ecological niche or the source community for most of the bacterial and viral pathogens that are causing respiratory infections in our community. It's the niche where they thrive, they can't exist without that niche, and from there on they circulate or spread within the community. And in general, we have a happy coexistence with most bugs around us. And it's actually only the last, I would say, 50 years that we start to think about how does that come? Because if those pathogens can be found in any healthy human body, why does one person not become ill and the other one does? Well, that's where this microbial community came into the equation and say, well, if there is an equilibrium in the microbial community on this mucosal surface, there's a state of resistance against acquisition, but mostly containment of bacteria and viruses passing by. Immune modulation, as we have just briefly discussed, and what we call resilience. So when you have a attack of a virus and you get a bit of a snotty nose, that might be good for the virus for spreading around the community. But once you have dealt with it, you pop back quite quickly into what we call eubiosis and keep on living. However, we think if that process does not work very well, it means that pathogens can actually overtake that niche, outgrow, invade, and disseminate through the body, and of course cause a lot of local or systemic inflammation. Okay, so that's the theory where we start by looking at the data. So what I've brought for you today is two studies that we have been part. One is looking at the microbiome at the time of quite significant respiratory infections. And then following, I'll move on to what happens preceding those infections to look at the physiology or pathophysiology of what seems to happen. So the first study I want to bring in here was executed by one of our former PhD students, who's now almost a licensed pediatrician, Wing Ho Man. And he executed and studied a very minutiously controlled case control study where we recruited 150 children between 0 and 6 years old with a community-acquired childhood pneumonia. It's a very wide, broad diagnosis, controlled in a 2-to-1 manner with children from their own community. And to be able to do that, we had those controls recruited and standby, I would say, in our database. They were just kept on going with their lives, of course. But at time that one case was admitted to the hospital, what we did was look in our database to see in our controls whether we had an age and gender matched child living in that area. We gave them a phone call and asked whether we could come within 24 to 48 hours to also obtain materials and ask some questions. So what that does is that it's creating a study that's very well controlled for circulating bacteria and viruses, which can be very seasonal and even change from year to year. So what we did with the samples obtained from cases and controls was we looked at a wide range of respiratory viruses by quantitative PCR, and we looked at the microbial community composition by 16S-based sequencing. Well, just in briefly, what does that encompass? Well, we take the samples from, as I said, the back wall of the nasopharyngeal wall. There's microbes there. We isolate their DNA of whatever is there, and we amplify a household gene, part of the 16S RNA gene, by universal primers. Basically, they fit in all bugs that are there. You don't need to know who's there. So you get all these amplicons from all these different bacteria. By sequencing those, you can say, okay, bacteria 1, 2, 3, 4 are there in that many copy numbers, which is a representation of the relative abundance in that upper respiratory tract. So that in a nutshell. So going back to our study, as said, we know that the back wall of the nasopharynx is the source community of pathogens, but we didn't know a lot what happened with those commensals, since we knew the environments were very different. So we had the idea that given the microbial community was theorized to overgrow and being aspirated and disseminated to the lungs, these communities at time of an acute community-acquired infection should be very similar. But the proof or the evidence was not completely there on community level. So this study was ideal because about 30 children were unfortunately that severe that they had to be admitted to the pediatric intensive care unit for support by ventilation, which required intubation of their lower respiratory tract. And what that gave us as an opportunity is that we could immediately take samples at the time they were intubated of upper and lower respiratory tract and compare the communities in both locations. Well, on group level, just looking at the color code, you see they are very similar. It is the nasopharynx of these 30 children, and this is the lower respiratory tract or lung community of these children. You see that there's slightly more diversity in the upper respiratory tract. So certain bacteria do not seem to hitchhike to the lungs, but on group level, otherwise it looks quite well. The good thing is we had paired samples. So when we looked at what we call the similarity between the community, the entire community in the upper respiratory tract and the lower respiratory tract, we saw very high similarity levels. This is the median with the spread. And if you look at what biological replicates from the same lungs, how similar they are, the Bogaert similarity index is around here. So you see that the similarity between upper and lower communities were actually approaching, sometimes even crossing biological replicates. And what made that for some children not the same was the absence of several of these very low abundant bacteria that did not seem to hitchhike to the lungs. And if you compare children's nasopharynx with the lungs of any other children, you see that that similarity is very low. So fortunately, we could prove that point and could move on. actually looking now at the nasopharynx, which is approachable in all these children where their lungs is not, if they're not ventilated, and compare the nasopharyngeal community of cases and controls. So if you first look at the characteristics of these children and their medical history, you already see that the cases had a much more common history of infectious problems, including wheezing, illness, and otitis, and even hospitalization for another lower respiratory tract infection, as well as they were significantly more often already treated with antibiotics in the past 6 months. If you looked at feeding, they were significantly less often breastfed for 3 months or more. And also looking at an environmental component, smoke exposure was significantly higher in those children, all risk factors which we know from literature, but we saw them as well in our community. So then if you look at those viruses that we found in those children, first of all, do we find any virus? Well, in cases that was nearly 100%, but maybe for some surprising, for me it was not anymore, but more than 80% of controls also have one or more viruses with them. As said, it can go very frequently hand in hand with just a healthy state. Then if you look at what types of viruses are present, we find very often human rhinovirus, a very common general respiratory virus, which interestingly enough was significantly less often found in the cases versus the controls. So it's very common in healthy people and less so in ill people. Well, RSV, respiratory syncytial virus, is very well known for its virulence in young children, and that was dramatically significantly associated with disease, but still there were kids having RSV in the control group as well. Human metapneumovirus is sort of a sibling of RSV. We saw the same significant difference. These 2 years that we recruited, there was hardly any influenza in the Netherlands where the study was executed, so we could not say anything about that, but all other viruses were basically equally found in cases and controls, and that was very reliable thanks to our very minutiously case control matched. So then if you look at the microbial community, I want to show you this plot, and that might sound a little bit, or that might look a bit complicated, but it's not really. What we try to do is look at the entire community, collapse that in a single dot into two-dimensional space. And basically, this is a sample, for example, that's depicted here. So any sample that is also depicted in this plot very close to that one has a very similar community profile. However, if it's plotted over here, it can't be more different, as are these dots on the other sides of this two-dimensional plot. And we superimposed the biomarker species driving the space here, where you see that gram-positive commensals are driving communities more on that side of the equation, Staphylococcus aureus more this side of the equation, whereas Streptococcus pneumoniae and Haemophilus influenzae, Moraxella, a little bit more that side of the equation. Well, we color-coded them, cases are red and controls are blue, and then you see sort of a standard deviation, standard variation around the median for these groups, which are significantly different. But of course, you immediately say to me, well, there's also huge overlap, and that's also not that surprisingly. So if you look at whether each of these data sets is predictive for infection or not, they're not performing so well. The viruses alone have a predictive value of about 0.7, or an area under the curve of 0.7, whereas the microbiota communities as a whole do a bit better. They are 76%, but the kids' characteristics, the clinical characteristics actually do even better, and you can't base treatment on that, can you? So then Wing Ho had this idea to try to combine these data sets and perform machine learning. So we could, without a primary hypothesis, see whether we could identify the key characteristics that were classifying those children. And to our honestly surprise, we found a very good classification model, which was very robust after cross-validation, where you found these 20 parameters with RSV as our number one, but then a group of bacteria that were either present or absent, being predictive of cases, and also some of the more patient characteristics which had a little bit less value. But overall, this model was able to classify 92% accurately, and that was for the whole group in general. If you stratify this over the common clinical phenotypes, pneumonia, bronchiolitis, mixed infection, or even virus-induced wheezes, which is more an asthma-like phenotype. And until recently, we didn't think that the microbial community was very much involved, or at least that it's not an infectious process. They all had a very good to quite good predictive capacity between 0.90 and 0.94. So you can imagine that this was for us a first hint to try to say, well, perhaps looking in this way to a microbial equilibrium might help us in a much more nuanced way with diagnosis and treatment. But we were a bit hesitant to publish these data because we were afraid people would take that signal as, oh, let's treat everybody with antibiotics. And that was now exactly not what we wanted to achieve. So making a long story short, what we also tried to do is see whether the microbial community at day of hospitalization, the first day, was predictive for duration of hospitalization as a proxy for severity of disease. And it was reasonably well predictive. And then someone said that part of these kids were treated with antibiotics. This is a true correlation. If you then treat it at day 1 with antibiotics, this correlation should be gone. And indeed, that was the case. If a child got antibiotics, the correlation totally disappeared, whereas it became stronger for all the children not treated with antibiotics, really supporting the idea that this microbial ecosystem is part of the pathophysiological process, but also of the process of natural recovery. And Emma de Koff even looked beyond in 4 to 8 weeks following. And what she even could do was say, well, once a child had recovered from its initial lower respiratory tract infection, what was the likelihood it became sick, and could we predict that with, again, baseline microbial community at day 1? And indeed, she found a relatively strong signal, and we could identify biomarkers that were driving the risk of recurrence of infection in the following 4 to 8 weeks. So just to give you a bit of an idea, that there seemed to be true to looking at a wider microbial community. And we had another paper 2 years before where we could not only associate the nasopharyngeal microbiota at times of RSV infection with disease, but also with grade of severity of disease. Okay. So that leaves me to look at the respiratory microbiome preceding respiratory infection. So in contrast to, actually nicely analogy to what we've heard in the previous talks, the microbial community of a baby can be regarded as an organ system, but this organ system will only start to develop from the moment a baby is born, although it really has to function quite well. So you can imagine it has to develop quite quickly. But because it's starting to develop at the time a baby is born, it's heavily influenced by environmental factors. You can imagine mode of delivery, siblings, antibiotic use, smoke exposure, etc. And in analogy to, for example, the gut microbiome, we thought that was very important for development and stability and resilience of that initial infant microbiome. So we designed a prospective study called the Microbiome Utrecht Infant Study, where we recruited during pregnancy, that allowed to take samples right after delivery to look not what was colonizing, but what was seeded on and in the baby. And then at day 1 week, 2 months, 1 and so on, to see how then the child assembled its own microbiome and developed this further. And we did that for multiple locations within the human microbiome. But of course, today I will only focus on the nasopharynx. So again, you have here one of these plots. Again, each dot represents a total community over time, and they're color-coded according to the day and week and month they were taken. And what you see is that basically all children follow a similar microbial community development in their respiratory tract. They're not different trajectories. However, we see that these confidence intervals are quite wide, especially in the middle. Some children arrived halfway this caterpillar at 2 weeks of life, and others only after 6 months of life. So we wanted to look at what was actually driving this community development. And in the very early first week, we saw that was mostly season of birth. That was whether you were breastfed with mode of delivery and antibiotic use, all quite logical, isn't it? And then if you looked further across the whole year, first year of life, you saw that the number, the most important ones were exposure to other people, having siblings sharing their microbiota, and daycare attendance, where of course lots of children are sharing their microbiota, and the other factors remained important throughout the first year of life. So then we tried to connect that with respiratory health, and this is a term, healthy born community. So what we did was looked at how frequently that they have respiratory infections combined with fever to be able to really separate those infections out. And this is the normal distribution you see here from 0 to 7 infections. So we stratified according to below average, average, and above average. And then we went back to this microbiome development, and we calculated the microbiome age versus chronological age. And for the green group, you see that's quite a relatively nice linear association. For the average infection group, it starts off quite linear and it starts to accelerate as soon as those first infections after the first 2 months of life start to pop in. But for this high risk group, we saw that actually the children deviated very quickly within the first 10 to 30 days away from the others. So the microbiome age was preceding their factual age. So if you then look at the trajectory, I would say nasal types that the children followed, you see a very typical development. And we were hypothesizing that the time of arrival in each of these profiles was associated with infection risk. So we calculated that, and indeed, if you arrived quite quickly in a gram-positive commensal community with Corynebacteria and Dolichogranum, which are lactic acid-producing bacteria, you actually were more likely to be in the 0 to 2 infection group. If you very quickly moved from that one to a Corynedola with more Xella predominance, that was not so beneficial, as was not when you arrived, the time to arrival in a Haemophilus-dominated community. And here this is confirmed for biomarker species only. So we also looked at, besides microbiota development, again, to viral presence, even during these asymptomatic routine sampling, as well as in a subset of children to the mucosal gene expression to see what was going hand in hand in host microbiota interaction. So we tried to get an idea about this triangle of interaction. So if you look at the viruses, a viral carriage, I would say in those children, what we basically saw that already quite early in life, some of these children in the first weeks or months of life were colonized or infected with viruses, especially human rhinovirus. But as we knew, they were not yet symptomatic. We only started to see the first symptomatic children from the age of 2-3 months of life onward. So we thought that's interesting. And basically we could prove that the first viral detection was significantly preceding the first respiratory tract infection reported. And if you looked at the first virus to first infection, it was always positive number, except for some that were coincidental, but never the other way around, suggesting that first viral detection was always preceding first symptomatic infection. So we said, well, let's focus on that. Is that relevant for these children? So we calculated time to first viral infection versus your risk, ultimately, of developing recurrent infections. And indeed, the group with 5 to 7 infections had a much significantly faster time to first asymptomatic viral detection compared to the other 2 groups. So then if you look at the mucosal gene expression development, you saw again a nice developmental pattern over time. And here you see the first 6 months of life. And at the beginning, you see that they all cluster, but then you see that some have a more rapid pace again of gene expression development and others a bit slower. And if you look at modules of genes that are co-expressed over time, you see that there's 2 modules standing out, module 1 and 3, that are very inflammation associated. The first one is TLR cascades and neutrophil activation, basically inflammasome activation, and module 3 was almost uniquely interferon signaling pathways. And you see that both are low expressed early in life. Here you see, probably as because of first exposure to microbes, a little glitch like, oh gosh, we need to respond, and it's dampened again. And only later on, this is upregulated. Whereas module 2 and module 4 were much more homeostasis and epithelial maturation, metabolism modules that were initially higher upregulated and then downregulated again. Well, I won't give you all the details, but we really tried to pair by pair module the correlations. And the overarching conclusion we came to was that if you had very early and therefore also prolonged Corynebacterium dolichogranum, again, these gram-positive lactic acid producing bugs in your nasopharynx, that was going hand in hand with more module 2 and 4, epithelial cell maturation and maintenance development. And this was going hand in hand with later first viral acquisition. Whereas if your viral acquisition was a lot earlier, that was going hand in hand with earlier and prolonged Moraxella, Haemophilus, and other gram-negative and all type of bacterial colonization. And that was also going hand in hand with earlier upregulation of those interferon signaling pathways and neutrophil activation and recruitment pathways, as well as other inflammasome-mediated pathways. And that triangle was related to more infections in the first year of life. And now we have followed this cohort until the age of 6. So now we will be able to look at long-term health, and we've been looking in more detail to atopic disease, as well as airway reversibility and symptoms of asthma, etc. So hopefully that will be the next stage of this cohort and this nice puzzle. So regarding biomechanisms, you probably will tell me, well, you don't show really mechanisms, you show more molecular epidemiological associations. Well, there's people who've done that, only showing data of one group that has used mouse models to test either this group of Corynebacteria or this group of dolichogranum and picarum in a mouse model challenging with RSV or RSV plus Streptococcus pneumoniae. And in general, what they found is if you treat those mice with our gram-positive commensal bacteria, that they have lower RSV replication, lower bacterial counts, and almost complete stop of invasion of pneumococcus and inflammation markers, including LDH and albumin, which are significantly lower if you pre-treat it, and also counts for dolichogranum and picarum, which was published last year by the same group. So that brings me in summary to that we try to look at the airway microbiome, the development, how the microbiome might be intermediate between risk factors and respiratory outcome. And we think that those data hopefully will help us with better diagnosis and hopefully more tailored treatment, but also in trying to see whether we could intervene early and prevent infections or recurrent infections. And how could you do that? Well, now we have a much focus on treating harmful bacteria and trying to eradicate these. But what we now see is that when these guys are taking over, the good bacteria are generally already low present. And with our antibiotics, for example, we even further reduce them. So we need to find ways to either add, restore, reconstitute, or nurture the good bacteria. And an alternative is trying not to give these bugs, for example, back, but to try to use natural substance or metabolites that these microbes use to inhibit those bacteria and viruses or block attachment, for example, and thereby you can leave the natural community to naturally restore. Of course, also, I need to acknowledge my own team, including people that already left the group, pursuing further pediatrics and GP. And I have also one of the people that's pursuing dermatology, so they're going all different routes. This is my current group, half of them in the Netherlands, half of them in Scotland, and my two dear colleagues who have helped me to co-organize the cohorts and that we were talking about, and others we were not able to talk about. And of course, our funders, and hope as well that I left a little time for questions.
Thank you, Professor Bogaert, for a very interesting talk. We have the Q&A open as well for attendees if you would like to ask any questions for Dr. Waters. Sorry, Dr. Bogaert, and also for our other speakers as part of the panel discussion. Nino, I can see that your hand is up. Would you like to come in?
Debbie, that was absolutely fascinating. And so are there interactions between the gut biome and the lung biome? And did you see whether basically if a person has all good bacteria, they're good both in the gut and the lungs, or is there a mismatch?
Yeah, I thank you for that question. It was one that we had for quite a long time, and it's very timely because 2 weeks ago we had a paper published that actually looked also in this cohort at the connection between nasopharynx, oropharynx, and gut microbiome to see, because we had a publication as well. I did not have time today for that, but looking at the gut microbiome of these children, we could also, to our surprise, tie that to risk of infections. And it puzzled us a bit, like that must mean that this is connected. This is not a coincidence. So what we did is try to look at a cross-human microbiome network to see whether we could identify features of the microbiome that could support the what you say, the hypothesis that these are connected or communicating. And we indeed could see that. So you have these very niche specific communities. You saw one of them, the nasopharyngeal community today, which is different from the oropharynx and different from the gut. But if you connect them, you see that not so much these niche specific biomarkers stand out, but you start to see a backbone of bacteria that are not per se niche specific, but that are connecting these communities. And we do see that that is establishing more rapid in those first month again in those children that ultimately seem to be more resilient or resistant against infections. And you also see that it's more stable. So it's earlier present and it remains stable over the first 6 months of life, which is a clue that there is indeed a better establishment of a cross niche or a human microbiome network. And you do see that the children that have more susceptibility already from very early on have a different network, which is not as strong connected and also not as stable. You see that they keep on looking for their backbone of their network. So that's definitely an idea to say, okay, if we want to look in the future supporting these children and their resistance against infections, we might want to focus a bit more on the human microbiome as a whole instead of on niche specific communities. You do see that again, a short chain fatty acid producing bacteria, for example, are part of the more stronger and stable networks. So, and also either in the respiratory tract or the gut, we see that they're also, for example, connected to better responses to vaccines. So there really seem to be a more healthy and better connected also to the immune system connected community. So it's interesting. Yeah.
I guess, sorry, following on from that question then, in terms of just seeding the nasal passage for healthy commensals, I guess the gut is, we all know that we can, eat more yogurt or give prebiotics and ingest those, but how do you like reseed the nasal passage, or how might we go about changing that?
Yeah, it's a big wish of us already for a couple of years to say we see those signals now everywhere. It's not only in our work, you see it in Australia, you see it in America, you see it in Africa. So these are really apparently core commensals of our body. So we would love to develop, for example, a product whereby you can give those bugs back when you start in a beneficiary. For example, if you have antibiotic treatment in the first week of life for suspected early onset neonatal sepsis, you just wipe away these bugs. That's known for bifidobacteria, but also for these guys in your respiratory tract. So we already applied multiple times for a grant, say we would love to give those bugs back. And although people find that a very intriguing idea, they're also quite reluctant because we work in young children. I think we should have started the other way around with adults and then work our way back to children. But because the signal is so strong in these young children, we would love to do that. But we need to find a way because we get funders interested, but they're all hesitant because it's a live biological product in babies, basically.
Sorry, Jan, John, sorry, do you want to come back in there?
Yeah, you know what? I couldn't stop asking a lot of questions, but Devi, fascinating. So it's of course very tempting to say, oh, we give them good bacteria, but do we know something about the role of, let's say, chemokines within the lungs that actually generate the right environment for this? You know, like basically not the bacteria, but what is the lung doing or the gut doing to provide a real environment?
Yeah, well, for bifidobacteria, for example, we know that you can reconstitute these. So premature children, for example, now are commonly receiving Bifidobacterium infantis when born prematurely. We know that they are eradicated with, well, either not getting them because of C-section birth or eradicated by antibiotics. And they give them now in many NICUs to prevent necrotizing enterocolitis. And it depends probably on the strain that's given, but it seems to work. So you can do that. So not per se that because the inflammation or the milieu is different, these can't engraft. And I think if you compare adults which already have their own microbiome and their interaction with their immune system, I think it's more difficult to give them other bacteria that we find more beneficial. You see that with fecal transplants, for example, that they won't last for very long. They might help you to get rid of C. diff, but they most of the time don't last, probably because the body is not its appropriate microbiome. But we think when you're really early in life, you still have that chance to affect natural development. The body is still open for its population process. So that's why we're so eager to try to see whether we can affect it, because we feel the potential is much better in early life than later in life. So that's why, but yeah, you can reconstitute. It's a matter of being early, I think, before the damage is done or the severe inflammation or chronic disease is done. And also bear in mind, these microbes do not colonize the lower respiratory tract. They colonize I would say they're the port d'entrée, or they're protecting the route of entrance. So that's even more intriguing, I think.
I guess I've another question that maybe kind of links all your work together. So in terms of inflammation, I know, Dr. Ramirez, you've done a little bit, a lot of work on sudden infant death syndrome. And one of the hypotheses behind sudden infant death syndrome is that these infants have had some exposure to inflammation that makes their more vulnerable to cardiorespiratory failure. So if you'd like to comment maybe on, you know, that whether it's a second hit or it's a vulnerability within the infant, or what's exactly happening. Yeah, sorry, thank you.
Yeah, it's a fascinating question. And I think, you know, that actually there are some genes that predispose children to sudden infant death syndrome, and they belong to a class of those that affect the immune system. So I think you basically need a strong immune system as part of your defenses, you know, the resilience that Debbie and also Jody talked about. And so I think ultimately we need to really understand the immune system, the neural system, and the microbiome, how they interact. I think this is really beautiful how this symposium highlights that.
Jody, I can see that your hand is up. Would you like to come in?
Yeah, I had a question for Debbie. So do you see any differences in like the health of the nasopharynx colonization if the mothers like breastfeed versus, you know, bottle feed? I know you mentioned a little bit about that in there, but I wanted you to expand a little more.
Yeah, definitely. So we do see that breastfeeding is related to microbiome development in the respiratory tract. And we also know from looking at the breast milk of these mothers themselves that, for example, the corynebacteria seem to originate from the breast milk. So there's a direct connection link there. And we're currently trying to look whether because we have the matched mothers and we obtained nasopharynx or pharynx breast milk skin samples and gut samples from these mothers and vaginal samples that we are now, one of our postdocs is trying to one by one trace what microbes might come from what niche in the mother. But you do see indeed a correlation, which is even quite niche specific. So for the nose nasopharynx, there is a link with the breast milk, for the gut as well, but not for the oral cavity, for example, which is interesting because the children take it through the mouth, but the microbes are not the same one. So it's interesting. Or of course, children regurgitate, so the corynebacteria might stick there. But yeah, you see there is even niche specificity there, but there is a strong correlation with breast milk for sure.
That's really interesting.
Just another thing that kind of came up in multiple talks, I guess, Dr. Waters, you had mentioned about sex differences, and that's central to your work. I was just kind of wondering, maybe is that seen in Dr. Bogaert's work or Dr. Ramirez? Do you see different patterns or different intrinsic properties? And then maybe a follow-up question is, there's obviously male vulnerability during development, but I guess what can we learn from the female resilience rather than focusing specifically on the male vulnerability?
Yeah, so one approach that we're taking is looking, at least from the microglial perspective, if we sequence the microglia from the male offspring spinal cord and then also the female offspring spinal cord, and we just look to see what genes are different in the males compared to in the GIH males compared to the normal control males, it's really kind of unimpressive. I think there's something like 12 or 13 genes that are different in the males. And so that was very puzzling to us for a long time because we're like, oh, the males are so messed up. How is it that only 12 genes are different? But then you look at the females and thousands of genes are changed in the female microglia. And so now we're in the process of trying to figure out, okay, what are those genes in the females? Like what categories do they fall in? How are they controlled transcriptionally? So that we can try to identify perhaps a pathway that the males are unable to elicit. And then we can hopefully pharmacologically or genetically manipulate that pathway so as to protect the male. So we're looking at exactly how you're suggesting, Fiona, to just try to see what is it that the females are doing to protect themselves and see if we can make the males do the same thing.
Dr. Bogaert, would you like to come in there?
Yeah, I was so intrigued by this data showed by Dr. Ramirez on the dysautonomy and the disconnect between the respiratory system and the cardiac system. And you just very briefly mentioned it, COVID, in the topic, and I was immediately intrigued, been interested and involved in treatment long COVID patients. And what we see there a lot is that irrespective of severity of their initial disease, you see people going on for long end of times. And I find tachycardias, breathlessness, especially exercise intolerance in people that are very athletic, part of that syndrome, if you may call it like that. And I saw your data and immediately thought, is this part of what we're looking at? Because there's still so little known, but you feel that it is an inflammation driven entity, but we can't make sense of it. And I was looking at your data, I thought this might be what's going on.
Now, I think you're hitting a very interesting point is, you have this center, the breathing center, the heart center, all of this is anatomically totally interacting and close by. And what we haven't really studied, but I know from work done by Chris Wilson, he's at Loma Linda, that if you have an inflammation in the lungs, within literally minutes via the vagal afferences, you also alter the central nervous system interaction right there. So I think, the link between what you're studying and what Jody studies is really ultimately all converging into these centers that try to organize it, because ultimately, the heart and the breathing have to work in sync, and this synchrony is happening exactly in these areas. And so I think there is definitely a link, and we're just starting to unravel this, and not only us. I mean, you have to see this is a whole community of people that now start to unravel how the vagal afferences the immune system, etc., interacts. And so, yeah, Fiona and David, my God, what a great symposium to bring everything together.
Yeah, no, that's great discussion. Thanks very much. I guess coming back to your talk, Dr. Ramirez, it's kind of obvious that the system is, our physiology is generally primed, and it's this inhibition that's really important. So like in terms of inflammation, it's the anti-inflammatory or the vagal communication that's really important for dampening down, but then our systems are ready to respond, you know, such that when they see an infection or, you know, when there's opportunistic bacteria arise or activate the system. I was just kind of wondering if you'd comment on that, so that, you know, how important that inhibition is, or, you know, maybe is that the key to understanding some of this? And then maybe, as you mentioned, maybe too much inhibition then, you know, results in things like apnea or Rett syndrome, for example, inhibition gone wrong, perhaps.
Fiona, look, you hit something very sensitive to me, to myself, because I was a believer of excitation is doing everything to one believer that inhibition is doing everything. It's really the balance between excitation, inhibition, and I think the more complex the system gets in the in vivo situation, the more inhibition is really the regulator of the frequency of the interactions, of how the afferences work. So I think you hit it right. It's the inhibition that shapes how the system becomes robust. You know, because we're also astonished how robust the respiratory system is. You know, like, I mean, if you would just depend on excitation, I think it would not be robust. But you have the inhibition that helps to generate the interaction between the population of inspiratory neurons and expiratory neurons. And only when you go into this whole emergency situation of gasping, you shut down the inhibition, and now you become like a ballistic system that has to regenerate by pacemakers to bring yourself up. And by the way, also not only the breathing system, but also the heart. But this is really the last stance to survive. But the robustness really depends on inhibition. So yeah, thanks for that question.
Can I maybe just come in with a question for Professor Bogaert? And I just related to the data that you showed in your earlier slides. I was kind of intrigued by, you showed that the children, I think, who were cases, they had lower levels of rhinovirus compared to the healthier subjects. I don't know much about rhinovirus, but I was wondering, is there something maybe with regards to the host response to rhinovirus, which may be beneficial, I guess, with regards to not acquiring respiratory infection, and maybe that not being exposed to rhinovirus may be a risk factor for respiratory infection?
Yeah, it's an interesting thought process. We are really still, we haven't figured it out yet, because if you look at those first viral acquisitions in those babies that are actually fully asymptomatic, it is mostly human rhinovirus. And we know that they're not symptomatic at that time, but they are more at risk of consecutive development within, I would say, 60 days of other infections, and they have also more infections. So we think it's the timing of that first acquisition. We do think, what we do see is if you have that rhinovirus, it's immediately associated with upregulation of the interferon pathway. And interestingly enough, it also stays a bit more upregulated, so that actually might prevent your consecutive infection. But we have to look in a larger cohort, which we hope to do in the coming years, to tease that out, because we have that the hypothesis like you, that this human rhinovirus might actually put you on the right path as long as it's on the right time. And what we do see is often that, for example, people with asthma, asthma attacks might be triggered by human rhinovirus. So often we say, well, it's the virus, but we don't think it's the virus per se. It's the either what Jyoti was looking at, epigenetics of the patient, so they are sort of over-responding to that trigger, rather than that it's the virus itself. So it's probably more imprinted in the host, and or the microbiota that we know now as well seem to be also triggering more inflammation. Because I briefly mentioned the RSV paper, and what we saw again was at time of RSV infection, they all had an RSV infection, but if you had more gram-negative flora in your upper respiratory tract, you had also more pro-inflammatory pathways, IL-1, IL-8, IL-7, Th17 pathways. They all had the very same range of interferon triggering, but they had a very different, more bacterial triggered pathways. And the higher those pathways were, the more severely these kids were ill. So I think you're right. I think it's environment rather than the virus itself for human rhinovirus. And perhaps even in early life, as long as the right timing, it might help you to be prepared for what's to come.
Thank you. I have lots more questions, but I think we're running out of time. We've just hit the hour. Fiona, what would you like to do?
I was going to ask about more, but that's okay. I was maybe we'll just finish up. I was kind of wondering about translational physiology. So I guess a lot of work we're doing is based in animals, and I guess there's sometimes a bias against animal or work that done in rodents or lower species in how translatable they are to the human situation. So I was just wondering, could you comment? I guess there's some limitations with some approaches, with brain slices, you don't have that vagal inhibition, or even in some anesthetized preparations with phlebotomy, again, we're lacking some of that inhibition that may not translate in vivo. Obviously, we've seen some in vivo work that do, I guess, reinstate or give us confidence that we can see the same thing in behaving animals. But I guess there's kind of a little bit of a barrier in terms of sometimes the work that we do as basic physiologists translating to the clinic, and even in terms of new pharmacotherapies in the neonatal ICU or in the pediatric setting have been very rare over the last number of years, and things like prenatal steroids are still number one, and caffeine post-natally, driving anti-inflammation and driving breathing to stabilize breathing. I was just kind of wondering, where do you think we're at in terms of maybe new therapies or new targets, and what might we do to overcome some of that translational difficulties or barriers?
I think ultimately the key is to have all the approaches, to be honest. I think, the in vitro has allowed us to really dissect network interaction at the single cell level, which is not so easy in vivo. But then you have to take a stance and see how does it relate to the whole animal. And then from there, you have to interact with the clinicians to really understand how is it in the human. And so I think it's really a call for these institutes where you go from the bench to the bed and back from the bed to bench. And I mean, a lot of the things, beautiful work that's done by Debbie or Jody is really like asking for more animal experiments to control, like what is the lung doing really to the microbiome? Like what is a sterile environment doing versus a real environment with other kids in the same room? I mean, you could do this all in mouse models. So I think that the key is to be interactive, and because every approach has limitations, be aware of these limits.
Dr. Waters or Dr. Bogaert, would you like to come in on that, and then we'll wrap up the session?
Yeah, I agree. I think we have in our environment pretty good access to children and children's studies, and that's what we try to do, and try to do them as good as possible. And that's also why we need other colleagues who can translate that into their animal models, for example. It's not a competition, it's working complementary. And I think that realization that we need to do it all together as a community, and from there on move forward, I think it's becoming more and more clear that that's the way forward to try to get to new interventions. I think it's the only way.
Yeah, I agree. But, we're trying to work with obstetricians here to, first of all, just even pay attention to, does the pregnant woman have sleep issues? Because that's not even a thing. Like most OBs don't even ask that. So, I think, again, that bidirectional communication between the basic scientists and the clinicians can help to, open those doors new therapies.